Genes provide information for building proteins. They don't, however, directly create proteins. The production of proteins is completed through two processes. And guess what? These are transcription and translation. But before we start, let me ask you a question. Have you ever had to transcribe something? Maybe someone left a message on your voicemail and you had to write it down on paper. Or maybe you took notes in class and rewrite them neatly to help you review, because that's what I do. So let's now proceed. As this example shows, transcription is a process in which information is rewritten. Transcription is something we do in our everyday lives, and it is also something our cells must do in a more specialized and narrowly defined way. In biology, transcription is the process of copying out the DNA sequence of a gene in the similar alphabet of RNA. And it says here that it is the first step in gene expression, and it involves copying a gene's DNA sequence to make an RNA. Next, transcription. It is the process by which RNA is made from a DNA template, as you can see in the picture. Its purpose is to read the DNA one gene at a time. Meaning, the goal of transcription is to make a RNA copy of a gene's DNA sequence. For a protein coding gene, the RNA copy or transcript carries information needed to build the polypeptide, which is protein or protein subunit. And RNA is assembled in the 5 to 3 direction. Transcription is performed by enzymes called RNA polymerases. And the transcription has 3 stages. These are initiation, elongation, and termination. It says here that transcription initiates with RNA polymerase binding to the promoter region in the DNA. The transcription binding to the promoter forms the transcription initiation complex, while elongation, during transcription, the RNA polymerase, after the initial abortive attempts, traverses the template strand of the DNA in 3 to 5 direction, producing a complementary RNA strand in 5 to 3 direction. Lastly, the transcription termination in prokaryotes can either be Rho independent, where a GC-rich hairpin loop is formed, for Rho dependent, where a protein factor Rho destabilizes the DNA to RNA interaction. Now, let's define a translation. But before that, did you know that translation literally means to carry across? That's what translation means. And here, as we can see, it is the process of translating the sequence of a messenger RNA or mRNA molecule to a sequence of amino acids during protein synthesis. What is being carried across is information that originally was in the genome, in DNA, then gets transcribed into messenger RNA. And then that information is translated from the messenger RNA to a protein. So we're talking the same information, but it's going from one form to another, a nucleic acid code to an amino acid code in a protein. The translation is done not in individual letters. That's what it means. So the genetic code describes the relationship between the sequence of base pairs in a gene and the corresponding amino acid sequence that encodes. In the cell cytoplasm, the ribosome reads the sequence of the mRNA in groups of 3 bases to assemble the protein. So now let's differentiate the transcription and translation. Here, the location of transcription is in nucleus, while translation is in cytoplasm. In initiation, the transcription occurs when RNA polymerase protein binds to the promoter and DNA and forms a transcription initiation complex. Promoter directs the exact location for the initiation of transcription. In translation, it occurs when ribosome subunits initiation factors and tRNA bind the mRNA near the AUG start codon. In termination, RNA transcript is released and polymerase detached from DNA. DNA rewinds itself into a double helix and is unaltered throughout this process. While in translation, When the ribosome encounters one of 3-step codons, it disassembles the ribosome and releases the polypeptide. Lastly, which is the elongation, RNA polymerase elongates in the 5 to 3 direction. The incoming so the explanation is already there. But in addition to that, did you know that the purpose of transcription is to make RNA copies of individual genes that the cell can use in the biochemistry? Katulad ng mga na-define natin about transcription kanina. And the purpose of translation is to synthesize proteins, which are used for millions of cellular functions. And we are now in the last slide. As you can see, the picture, nakikita natin dito yung process ng transcription and translation. Transcription and translation take the information in DNA and use it to produce proteins. Let us always remember that. Transcription uses a strand of DNA as a template to build a molecule called RNA. The RNA molecule is the link between DNA and the production of proteins. During translation, the RNA molecule created in the transcription process delivers information from the DNA to the protein building machine. So these two processes are really essential for life. They are found in all organisms, whether in eukaryotic or prokaryotic. Converting genetic information into proteins has kept life in existence for billions of years. That's all.